trying to incorporate AI methods in every single drug discovery project in the company. We are basically mostly working on biologics development. So basically that's antibody design from early stages to preclinical stages. So in all these stages we have different kind of set of tools and methods we're trying to use. We are incorporating these tools into a platform we call Lab in the Loop. And that's a combination of different AI systems and methods, starting from generative AI models that are basically proposing all sorts of variants of a molecule, usually starting from some kind of a seed molecule that we're trying to improve. And then we're using these proposals generated by these models, feed them into the in silico predictors. Then we get some predictions that we can use for ranking these variants. We also take care of the multi-objective optimization. That means we looked at actually multiple different properties that are very relevant for drug development, which includes usually affinity, developability properties, to rank the designs, and then the top clones or the top basically variants of the molecule we send to the wet lab for the experimental characterization. And then from there we kind of close the loop by feeding these experimentally annotated molecules back into the models, and then the cycle repeats. And that's sort of the thing we call Lab in the Loop. The way how we approach the portfolio is by bringing that whole machinery into every single project in the company. Traditional way of developing drugs is usually you do the development in sequential stages. You are first screening for some function and affinity from, let's say, initial discovery campaign, and that could be, let's say, some library design or with display technology or some immunization campaign. And then you're basically screening for just affinity maybe and potency. And then the top performing clones, depending of, let's say, because we are working in biologics, mostly we are getting actually these molecules from animals. So you need to humanize them. And then there is another step of like sort of corrections that needs to be introduced into these molecules that often kind of introduces other errors in the molecules. And because of that sequential process of doing things one by one, right, we often kind of end up with a molecule that is not fully optimized because we didn't look at these properties all at the same time. And that's why like AI kind of, we believe, could really help, because we can actually in an initial stage and a discovery stage, we'll look at all of these properties at the same time with our in silico based predictors, or as we also like to call them, pseudo oracles. And then we try to kind of optimize the space based on the performances from these models. It's faster, certainly, than a traditional way. Of course, there are drawbacks, right? Not every model is like seeing a lot of data, for example. So there is still some uncertainty in the prediction that we need to take into account every time. Sometimes we don't have enough data to train a model that can, let's say, generalize across different projects. So you need to train these models from project to project, basically. So some of them are like project dependent, right? And so there's quite a lot of stuff that kind of, depending on the project, we need to adjust. Mostly I worked in academia, but I did work on proteins a lot. And then I started more of a generative AI models and the work on generative AI models. And then during COVID times, it was either writing another paper or starting a company. And because we were all working remotely, we kind of needed another interesting project, so we started a company. And only in a few months, we already had a bunch of VCs ready for the seed round and everything. But then among some of the potential partners, we had Genentech, who was actually very much interested in acquiring us. Since January until, let's say, August, we were already part of the bigger company, and we completed that acquisition process. So we had something that would be probably considered maybe fastest exit in the history of any startup. But certainly the things changed for us a little bit, because we wanted to be maybe a little bit of an AI first company. And then now we're part of a much bigger biotech pharma company where things are done in a bit different way. So we're finding different ways of how to incorporate AI into the existing system and try to kind of a little bit change or revolutionize the way how things are done in the company, how drug discovery is done in the company. A lot of things is not only about just bringing the tools and the methods and applying them. So we have to do a lot of stuff on the infrastructure, on the data side. Actually bringing these tools also changed a little bit the way how things are done in the company, like the culture of how data is stored, how data is generated. For example, a bigger focus on the negative data points that are extremely useful for AI systems. So it was, a lot of stuff that are kind of still in the process, but are happening in parallel with this, because it's, I think, very important we kind of develop all these aspects so that AI can be very successful in these projects.